Good day, class. In this lesson, we are going to talk about gametogenesis and fertilization. I believe these two processes are not entirely new to you. You have discussed them perhaps during your first year in college when you took your anatomy and physiology subject. But as a sort of review, let's define these two words. Gametogenesis is the process in which gametes are formed. In human males, the gametes are called sperm cells. In human females, the gametes are called egg cells or ova. Now, these gametes are produced using meiosis. Again, in meiosis, which we have discussed previously, a diploid cell, for the case of humans, a diploid cell means a cell containing 46 chromosomes. So, again, in meiosis, a diploid cell enters meiosis 1, producing two daughter cells which are now haploid. Meaning to say, they contain only 23 chromosomes. Kaya nga tinawag yung meiosis na reductional division. Then, in meiosis 2, each of those haploid daughter cells undergoes another round of cell division, producing in total four daughter cells from the original diploid cell that entered meiosis 1. So that's for gametogenesis. Now, let us take note that the product of meiosis are haploid cells. They either contain the maternal set of chromosomes or the paternal set of chromosomes. During fertilization, the process where the gametes unite, a single cell is formed and it is called a zygote. Unlike the gametes, which are haploid, the zygote is diploid. The fusion of the sperm cell and the egg cell restores the diploid number of chromosomes, which is 46 in the case of humans. In this lesson, we are going to discuss gametogenesis and fertilization on a more detailed and more technical standpoint. So I hope you brace yourselves for a lot of things that you are going to learn, a lot of words, new words that you will need to memorize. So come on, let's enjoy learning. I am presenting to you now the objectives of this lesson according to your syllabus. So at the end of this lesson, you should be able to explain the prenatal and postnatal life and the periods of development. But actually, let's forego the postnatal part. We will focus more actually on the prenatal part. The number 2, differentiate. You should be able to differentiate the terms commonly employed in the study of gametogenesis and fertilization. Number 3, differentiate human ovum and sperm in terms of their significance and morphology and development. 4th, explain how the body regulates reproduction. 5th, enumerate the hormones required in the regulation of reproduction. 6th, explain the significance of meiosis in gametogenesis and fertilization. And number 7, differentiate the stages of fertilization and prenatal development. So let's begin. Are you ready? Are you excited? Previously, we mentioned the two main purposes of cell division as enumerated by Bruker. Number 1 is reproduction, which can be either asexual or sexual, and number 2 is multicellularity. We also established that multicellularity is achieved by mitosis. Asexual reproduction is also achieved by mitosis. Asexual reproduction again is the process in which the descendants are genetically identical to each other and to their mother cell. Again, assuming that no mutation has happened during the process. But what is more relevant to our discussion now is the reproduction by sexual means, because this involves meiosis, not mitosis. During sexual reproduction, gametes are made that contain half the amount of an organism's genetic material. For example, for humans, the diploid number is 46. The cell that enters meiosis 1 initially contains 46 chromosomes, and then the gametes that are so produced contain only 23 chromosomes. Sa katunayan, bago pa man na-elucidate ni na Oscar Hertwig and Theodore Boveri ang mga kaganapan during meiosis, na-predict na ni August Weismann na nangyayari nga itong phenomenon na ito, which is reductional division, which we now call meiosis. Now, let's review meiosis muna before we proceed to gametogenesis para fresh pa sa sa brain. So again, meiosis is composed of two rounds of divisions, meiosis 1 and meiosis 2. Yung pumapasok, yung cell na isinasalang sa meiosis 1, that is a diploid cell containing 46 chromosomes. Papasok siya ngayon sa prophase 1. In meiosis 1, the prophase here is divided into 5 stages. In the proper order, it is leptonema, zygonema, pachinema, diplonema, and diakinesis. So in leptonema, ang nangyari doon is nagko-condense yung chromosome. And this process is similar to the prophase of mitosis. So ayun, nagko-condense yung chromosome. Then during zygonema, nagsisimulang mag-form yung yung tinatawag natin na synaptonymal complex in between the sister chromatids of the homologous chromosomes. The process of synaptonymal complex formation is called synapsis. And then, during pachinema naman, natatapos yung proseso ng synapsis, nagiging connected na yung dalawang chromosomes, yung homologous chromosomes from telomere to telomere, meaning from end to end. It is also during pachinema where crossing over happens. When you say crossing over, nagpapalitan ng segments yung dalawang chromosomes na homologous sa isa't isa. Then during diplonema, nagsisimulang maghiwalay yung homologous chromosomes, and that is possible because of the breaking down of the synaptonymal complex. Then during diakinesis, separate na talaga sila, yung dalawang homologous chromosomes na yon. After diakinesis, papasok na ngayon sa prometaphase 1. During prometaphase 1, similar sa prometaphase ng mitosis, yung kinetochore microtubule ay magko-connect sa mga chromosome. So, ang difference lang nito sa prometaphase ng mitosis is doon sa mitosis, bawat isang chromosome ay maa-attach sa dalawang kinetochore microtubules, one coming from each pole of the cell. Dito naman sa meiosis 1, bawat isang chromosome ay maa-attach lamang sa iisang microtubule, iisang kinetochore microtubule lamang, coming from either of the poles. And then, also take note na dito sa prometaphase 1, the chromosomes are, hindi pa sila naka-arrange along the equatorial plate or metaphase plate. Instead, they appear as if they are disorganized sa region ng nucleus. Also, dito sa prometaphase 1, the nuclear membrane is already degraded. I forgot to mention kanina, but by the end of prophase, sa bandang diakinesis, ay nag-breakdown na din yung nuclear membrane. Again, bakit kailangan mag-breakdown ng nuclear membrane? Because the kinetochore microtubules have to gain access to the kinetochores of the chromosomes. During metaphase 1, nag-a-align yung mga chromosomes, but ang difference nito sa metaphase ng mitosis, doon sa mitosis, isang layer lang ng chromosomes yung na-form along the metaphase plate. Dito naman sa metaphase 1, dalawang layers ng chromosomes ang na-form. And I would also like to mention again na nangyayari yung tinatawag na independent assortment. Ibig sabihin, if sa isang layer nandoon yung maternal chromosome, that doesn't mean na all chromosomes in that layer are maternal. So combination siya. Sa isang layer may maternal, meron ding paternal chromosomes. The chromosomes from the maternal side and from the paternal side don't necessarily have to be located in one layer. So that's what it means to say independent assortment. And then during anaphase 1, the homologous chromosomes are separated from one another. So what is pulled towards one end or what is pulled toward one pole of the cell is a copy of a chromosome, and that copy of the chromosome comes in the form of dyad. Again, when we say dyad, it is composed of two sister chromatids or two monads. So, in the case of humans, meron tayong 46 chromosome. Ibig sabihin, what comes towards the direction of one pole ay 23 chromosomes. But each of those 23 chromosomes are in the form of dyads. Then during telophase, magre-reform yung nucleus. Yung 23 chromosomes na iyon, in the case of human, will be wrapped around the nucleus. So dalawang nucleus ang magpo-form. Then telophase is also happening concurrently with cytokinesis. So, ibig sabihin sa cytokinesis, maghihiwalay naman yung cytoplasm. In animals, such as humans, cytokinesis happens with the help of the contractile proteins underneath the plasma membrane, yung mga actin filaments and myosin filaments. So, they will contract and then they will form the cleavage furrow. Sa plants naman, di ba, I mentioned, as we discussed, sa plants, cytokinesis happens by the deposition of cell wall material along the middle of the cell. Then, so yun, the products of meiosis 1 are two cells that are already haploid. Now, each of those haploid cells will undergo meiosis 2. Yung meiosis 2, ito talaga yung stage ng meiosis na mas similar talaga siya sa mitosis. So, what happens in prophase 2 ay magbe-break down yung nuclear membrane na na-form during telophase 1. So, magbe-break down yung nuclear membrane, then the kinetochore microtubules will have an access again to the kinetochores. Then, in prometaphase 2, so ayun, nakakonect na yung mga kinetochore microtubules sa kinetochore. This time, bawat isang chromosome, sa 23 chromosomes per cell, bawat isa sa kanila ay makokonect sa dalawang kinetochore microtubule, one coming from each pole of the cell. So, di ba, sabi ko nga, similar na siya sa mitosis kasi dalawang kinetochore microtubule na ang mag-a-attach sa bawat kinetochore ng bawat chromosome. Then, in metaphase 2, similar again sa mitosis, isang layer lang ng chromosomes ang i-form nila along the equatorial plate or metaphase plate. Then, in anaphase 2, again, similar ulit sa mitosis, the chromosome being composed of two sister chromatids will be separated from one another. And the molecular mechanism behind this ay na-discuss ko rin sa inyo, di ba? Yung the ones involving cohesins, separase, ubiquitin, and the anaphase promoting complex, also known as cyclosome. Na-discuss ko din to sa inyo. Then, sa telophase 2 naman, magpo-form ulit yung nuclear membrane to encapsulate the chromosomes. So, so the products of meiosis 1 are haploid cells. Then the products of meiosis 2 are also haploid cells. Then you will ask me, Sir, bakit haploid pa rin yung product ng meiosis 2? Bakit hindi na siya, if 23 chromosomes yung haploid number, so bakit by the end of meiosis 2, hindi siya naging, 23 divided by 2? That's 11.5. Bakit hindi 11.5 chromosomes yung mayroon ang bawat cell na nagpo-produce ng meiosis 2? That is because yung sa meiosis 1, yes, haploid yung mga products, haploid cells yung products ng meiosis 1, but each chromosome kasi ng haploid cells na iyon ay composed of two sister chromatids. Each chromosome is in the form of a dyad. Dito naman sa meiosis 2, haploid din yung cells na napoproduce, but each of the chromosomes of those haploid cells are in the form of monads. So, isang chromatid lang. So, yun yung difference nila. Ayan, showing you the karyotypes of a diploid cell and a haploid cell. So, nasa left banda ng slide, you can see the karyotype, a sample karyotype of a diploid cell. Each chromosome that you can see is in the form of a dyad. Some obvious things that I want to point out here is yung sex chromosomes ng person consists of an X chromosome and a Y chromosome, telling us that the source of the specimen used to produce this karyotype is a male individual. Another thing that I want to point out is from chromosomes 1 to 23, normal naman yung number. Tagdadalawa per chromosome number. Sa right naman na banda ng slide, showing you the karyotype of a haploid cell. So, ayan, paki-notice, yung chromosome 23 niya consisting only of one chromosome, and that is an X chromosome. Alternatively, that can also be that can also be a Y chromosome. And then, yun, paki-notice niyo, from chromosome 1 to 23, isang chromosome na lang, wala na siyang partner. So, ganyan ang itsura ng haplotype, ng karyotype kapag haploid yung cell. Ayan, so finally, we're done with our review for meiosis and the karyotype. Let's now talk about gametogenesis proper. Relevant to the discussion of gametogenesis are two terms, isogamy and heterogamy. When we say isogamy, ito yung phenomenon kung saan yung dalawang gametes na pinoproduce ng isang organism cannot be distinguished from one another on the basis of morphology. Iso meaning the same. Example nito ay sa isang fungus species na pinapakita sa slide. So as you can see, the gametes that are formed are not labeled as egg cell or sperm cell. Instead, they are labeled with a positive and negative symbol to denote that they are different biochemically, but we cannot distinguish them from one another on the basis of their morphology. So nangyayari naman to sa fungus and sa plants, sa algae at sa fungus and sa algae. But more commonly, in nature, the two gametes that are produced by an organism are actually different in terms of their morphology. And in that case, we call the phenomenon heterogamy. And we describe the organism as being heterogamous. Pag isogamy naman, yung adjective form niya ay isogamous. For example, sa atin, sa humans, the sperm cell is very small when you compare it to the size of the egg cell. So, napakahigante ng egg cell in comparison to the sperm cell. Another na unique na characteristic ng sperm cell aside sa kanyang napakaliit na size ay mayroon siyang isang flagellum, the structure that propels it so that the sperm cell is able to reach the ovum for it to be able to fertilize the ovum. Yung ovum naman natin, wala siyang special structure like the flagellum that will make it that will render it motile kasi yung gagawin lang naman ni egg cell is maghihintay sa darating na sperm cell. But what is special about the egg cell is yung napakalaki niyang size, that is because of the abundance of cytoplasm. And that cytoplasm contains the nutrients. Because yung egg cell kasi, siya yung magsusupply ng nutrients that will nourish the developing embryo soon. No? Yung samantalang yung yung sperm cell naman, mostly yung contribution niya lang naman talaga to the zygote and to the embryo is yung genetic material contained in the sperm cell. But yung egg cell, yung kino-contribute niya aside sa genetic material na mayroon siya is yung nutrients provided or present in the cytoplasm of the egg cell. We can also observe some variations in other organisms. For example, may iba na yung sperm cell nila ay mayroong more than one na flagellum. Let's have a closer look of our human gametes. Look at the left side of the slide, showing you the structures of the mature human sperm cell. It consists of a head, a midpiece, and a tail. It is what are present in the head? The nucleus, at itong isang special na organelle, we call it an acrosome. The acrosome is a modified organelle. It consists of enzymes that are needed by the cell, that are needed by the sperm cell, so that the sperm cell is able to digest certain substances that will allow it to fertilize the ovum. Yung ovum kasi, class, mayroon siyang mga barriers, no, that will hinder the fusion of the sperm cell with the ovum. But because of the presence of these enzymes contained in the acrosome, then the sperm cell is able to digest those obstacles. So, ayun, makaka-fuse na siya with the egg cell. The midpiece of the sperm cell is packed with microvilli. You know that the sperm cell has to propel itself through the female reproductive tract to reach the ovum. So for that to be possible, yung pag-move kasi ng flagella, class, is an active process. It requires ATP. And you know that the mitochondria are the organelles that are responsible for the production of much of our ATP. So you learn in biochem, di ba, na-discuss niyo sa biochem yung glycolysis, yung Krebs cycle, and then ultimately the oxidative phosphorylation. yung Krebs cycle and yung glycolysis, kaunting ATP lang yung pinoproduce nila. But they produce also, aside from ATP, they produce NADH and FADH2. Now, those NADH and FADH2 become the fuel of oxidative phosphorylation. Ito, reveal to sa yung biochemistry. Huwag matakot. So that your mitochondria, by way of oxidative phosphorylation, is able to produce large amounts of ATP. Now, those ATP become the fuel for the movement of the flagella. Next is the flagella or the tail. And ito na nga yung ginagamit ng sperm cell to swim. And the next is your ovum. Yun nga, sinabi ko kanina, wala namang masyadong special sa kanya maliban sa kanyang giant na size. And the purpose is to provide most of the nutrients for the developing embryo later. You might be wondering kung ano yung polar body. We will discuss that later na lang. The process of sperm cell production is called spermatogenesis, and the process of ova or egg cell production is called oogenesis. These processes involve meiosis. Previously, we established that in meiosis, 4 haploid cells are produced at the end of meiosis 2. But in animals, spermatogenesis, yes, it produces 4 haploid sperm cells, but oogenesis produces only a single haploid egg cell. Hindi 4, but just a single haploid egg cell. So, bakit kaya isa lang? Bakit hindi katulad sa spermatogenesis na 4 yung napoproduce? To know the answer, stay tuned lang sa ating video, no? But before that, let us begin discussing the spermatogenesis. Spermatogenesis happens inside our organs called testicles or testes. Inside the testes, you have these structures, ito mga kulot-kulot na mga tubo. We call them seminiferous tubules, which you have discussed, I believe, in your histology laboratory. These are the important resident cells of the seminiferous tubules. Number 1 are the Leydig cells, also known as the interstitial cells. Mayroong magsistimulate sa kanila, mamaya pag-uusapan natin kung ano yon. But upon being stimulated, yung response ng Leydig cells is magpuproduce sila ng hormone na tinatawag na testosterone, which is especially high in males. One of the functions of testosterone is to promote spermatogenesis. Kaya naman kailangan talaga na napoproduce yung testosterone sa location na ito, no, ng testes, kung saan pinoproduce yung mga sperm cell. Now, another resident cell of the seminiferous tubules are the Sertoli cells or the sustentacular cells. Among other functions, the Sertoli cells physically and nutritionally support the developing sperm cells. This is another illustration from Tortora. You can see the green cells there. They are the interstitial cells or the Leydig cells. Once again, they produce testosterone. Pasensya na sa mga paulit-ulit. These repetitions help you memorize things. Then another character dito sa seminiferous tubules are the sustentacular cells or the Sertoli cells, which support the development of the sperm cells. And then finally, yung talagang main character, yung bida talaga dito sa seminiferous tubules are the developing sperm cells. Now, ito na talaga yung proper discussion na talaga natin for spermatogenesis. We are now going to talk about the different stages of sperm cell production. So, in the seminiferous tubules, you have these stem cells called spermatogonia. So, sa brutal nyo, yung nakasabi lang siguro ata doon is spermatogonia. But in your home kera, na-identify yung dalawang types ng spermatogonia, type A and type B. Now, let's begin with type A spermatogonia, which are stem cells. Ang ginagawa ng type A spermatogonia ng mga stem cells na ito is nag-undergo sila ng mitosis, which is non-reductional. Remember, in mitosis, genetically identical yung cells na pinoproduce. So, ayun, mag-undergo siya ng mitosis, producing daughter cells. Sa dalawang daughter cells na yon, yung isa ay mananatili siyang type A spermatogonium na stem cell. Mayroong matitirang isa to keep the cell line intact so that hindi mauubos, hindi makukonsume yung supply ng stem cell. So, kailangan mayroong matirang stem cell. But the other daughter cell is the one that will experience cell differentiation. Siya yung papasok ngayon sa succeeding stages of differentiation that will ultimately end up in producing a mature sperm cell. Now, that type A spermatogonium will undergo a series of mitotic divisions. Take note ha, mitotic, hindi pa meiosis, mitotic pa. Producing several type A spermatogonia pa rin, but not anymore stem cells, but progenitor cells. And then ultimately, yung mga type A spermatogonia na progenitor cells will undergo mitotic division again to produce type B spermatogonia. So, again, type B spermatogonia. Those type B spermatogonia are the ones that will undergo mitosis, to produce yung cell naman na tinatawag na primary spermatocyte. Yung primary spermatocyte, which is diploid, diploid pa siya kasi yung process na nag-produce sa kanya is mitosis. So, diploid pa siya, siya yung papasok, siya yung isasalang sa meiosis 1. So, ayun, mag-undergo siya ng meiosis 1. By the end of telophase 1, makakaproduce siya, by the end of telophase 1 followed by cytokinesis, makakaproduce siya ng dalawang secondary spermatocyte. Okay, tanong ko para sa inyo. Yung secondary spermatocyte, ano yan siya? Diploid or haploid? Sagot. The answer is haploid kasi product siya ng meiotic division. So, haploid. But again, bawat chromosome niya is composed of 2 sister chromatids. So, yung mga chromosome na mayroon siya, we call them dyads. Next step, bawat isa sa secondary spermatocyte na iyon ay mag-undergo naman ng meiosis 2, ending up with the production of 4 spermatids. And then, each of those 4 spermatids will undergo a process called spermiogenesis. Spermiogenesis is the maturation of the spermatids into sperm cells or spermatozoa. Yung spermatozoa, sila na talaga yung mayroong tail. Yung spermatids kasi wala pa silang tail. So during the process of spermiogenesis, they will acquire or develop their tail, which is flagellum technically. Ito naman, another illustration. Uulitin ko lang basically yung mga pinagsasabi ko kanina para mas tumatak sa isipan. So, we begin with a spermatogonium. Let's say that spermatogonium contains 4 chromosomes. The 4 chromosome, yan yung diploid na number niya, 4. Then, yung spermatogonium na iyon ay magpo-produce ng primary spermatocytes, which are also diploid cells. Then, pinakita sa illustration na yung primary spermatocyte ay nag-duplicate na siya or nag-replicate na siya ng kanyang DNA, although hindi pinakita sa illustration na ito na nag-undergo siya ng crossing over. But in reality, nagko-crossing over talaga yan siya. Then, by the end of the first meiotic division, nakaproduce na siya ngayon ng dalawang secondary spermatocytes, both of which are haploid. Kita nyo naman, each secondary spermatocyte contains 2 chromosomes na lang. But each of those 2 chromosomes comes in the form of a dyad. So kahit dalawang chromosomes na lang yung mayroon siya, in total, 4 sister chromatids talaga ang mayroon siya. Next in the series, each of those secondary spermatocytes will undergo meiosis 2 or the second meiotic division. By the end of this process, bawat isang secondary spermatocyte ay nakaproduce na ngayon ng dalawang spermatids. So in total, ilang spermatids ang na-produce coming from the initial primary spermatocyte? The answer is 4. Now, each of those spermatids are still haploid kagaya ng secondary spermatocyte, but this time, the chromosomes of the spermatids consist of only one monad. They do not anymore come in the form of dyad, but monad na lang. So, bawat spermatid in the illustration ay mayroong dalawang chromosome. How about chromatid? Ilang chromatid? Dalawa rin. Yung secondary spermatocyte, dalawang chromosome but 4 na chromatids or 4 na monads. And then finally, yung mga spermatids na iyon ay magmamature into the spermatozoa or sperm cells by a process called spermiogenesis. Now, yung illustrations na pinakita ko so far ay napakasimple lamang, but in reality, things are actually more complicated. In reality kasi, class, yung mga spermatogonia, yung mga type A spermatogonia, and then later yung mga spermatocytes and spermatids na na-produce, they are actually not produced in a typical way. May something na unique sa kanilang cell division. Can you identify what that is just by looking at the illustration provided by Junquera? Can you? Okay, the answer is incomplete yung cytokinesis nila. Notice the illustration here. The cells produced, the spermatogonia and the spermatocytes are not completely separated from one another after telophase. Hindi kompleto yung cytokinesis nila. Instead, each of the cells are actually physically connected by what we call cytoplasmic bridges. Now, what do you think is the purpose of the cytoplasmic bridges? The answer is to synchronize the spermatogenesis. By way of these cytoplasmic bridges, the cells are able to communicate with one another directly so that sabay-sabay sila na nagmamature. Okay, ulit. The spermatogonia produce two cells. One of the daughter cells will replenish the stem cell pool, and then the other goes through a series of mitotic divisions to produce the type A spermatogonia na progenitor cells, which in turn also undergo mitosis to produce the type B spermatogonia. The type B spermatogonia will undergo mitosis again. Again ha, mitosis pa rin. Mitosis yung i-undergo ng type B spermatogonia to produce primary spermatocytes. And then, these primary spermatocytes are the ones that will undergo the first meiotic division, producing two secondary spermatocytes. Dalawang secondary spermatocyte per each primary spermatocyte. But since in the illustration, dalawang primary spermatocyte yung pinakita ni Junquera, that's why apat in total yung secondary spermatocyte na pinakita niya rin. Each of those secondary spermatocytes, which are by the way haploid, will undergo the second meiotic division to produce another batch of haploid cells, which we call the spermatids. And then the spermatids will undergo a maturation process called spermiogenesis to produce the mature spermatozoa and also producing residual bodies. Residual bodies are the extra cytoplasm coming from the spermatids. Notice that in the illustration na yung spermatids ay malalaki and they have excess cytoplasm, whereas yung mga mature na spermatocytes are maliliit. That's because during the process of spermiogenesis, they lose certain packets of cytoplasm, which are called the residual bodies. Now, question, what happens to these residual bodies? These will be phagocytized by the sustentacular or Sertoli cells. And finally, we're done with spermatogenesis. Dito naman tayo sa production ng egg cell, which is called oogenesis. If spermatogenesis happens in the testes, oogenesis naman happens inside the ovaries of the females. Yung meiotic division na nangyayari during oogenesis is somewhat different from that which happens during spermatogenesis. Sundan ang illustration. Let us begin with a primary oocyte which is diploid, just like the primary spermatocyte. The primary spermatocytes enters meiosis 1 to produce two cells that are haploid. However, those two cells are different in terms of their size. One daughter cell is called the secondary oocyte and the other daughter cell is called the first polar body. While each of the two cells essentially receive the same amount of genetic material, however, it is the secondary oocyte that receives much of the cytoplasm from the original primary oocyte. Essentially, the first polar body is just like, para lang siyang discard material. Parang basurahan lang talaga yung polar body. Yung composition kasi ng polar body is only the nucleus containing the 23 chromosomes that are in the form of a dyad and just a very little amount of cytoplasm and then the plasma membrane. The goal really of producing the first polar body is simply just to discard the 23 chromosomes. The first polar body may or may not undergo meiosis 2. If nag-undergo man siya ng meiosis 2, yung daughter cells na maproproduce ng first polar body will also be polar bodies. Yung secondary oocyte naman, it will also undergo meiosis 2. After meiosis 2, dalawang cells yung mapoproduce niya. Yung isa malaki, yung isa naman polar body rin, which we will call the secondary or the second polar body. So, ibig sabihin, kung nag-decide, kung yung first polar body man is able to undergo meiosis 2, question, ilang polar bodies in total ang na-produce at the end of meiosis 2? The answer is 3. Because the first polar body will produce 2 polar bodies, then yung secondary oocyte will produce another polar body. So 3 na in total. But if yung first polar body ay hindi nag-undergo ng meiosis 2, ibig sabihin, at the end of the process, dalawang polar bodies lang yung na-produce mo. Which is, yung first polar body, which is produced by meiosis 1, and then yung second polar body, which is produced by meiosis 2. I hope that is clear. Ulitin natin para mas maintindihan. In this illustration, nagpakita siya ng isang oogonium, which is yung sperm cell. Ito yung counterpart ng spermatogonium sa babae. Yung oogonium, mag-undergo siya ng DNA replication as a preparation for mitosis. Then magpuproduce siya ng primary oocyte, which is also diploid. Yung oogonium, diploid din yon. Yung primary oocyte, isasalang sa first meiotic division, producing 2 haploid cells. Yung isa malaki, we call that the secondary oocyte. Yung isa naman maliit, which is called the first polar body. Yung secondary oocyte ay mag-undergo ng second meiotic division to produce one large cell and another polar body. Sa illustration na ito, pinakita na yung first polar body ay nag-produce ng dalawa pang polar bodies. So, at the end of the second meiotic division, 3 polar bodies yung na-produce plus isang malaking cell, which is the ovum. Now, ulitin natin. Guys, if medyo nagsawa na kayo sa boses ko dahil sa paulit-ulit, you can just skip to the later parts of the video. I am intentionally repeating my explanations using different illustrations para klaro sa iba. Okay? Okay. This time pala, let's make things a little more complicated. So again, we begin with an oogonium. Did you know that pinagbubuntis pa lang ng lola mo, yung mama mo, ay yung oogonium na magiging ikaw ay nandoon ka na sa mama mo na fetus pa lang. No? It's amazing. It's so in a way parang pinagbubuntis ka na rin ng lola mo. Sabay kayo ng mama mo na pinagbubuntis ng lola mo. So it's amazing, no? Next, yung oogonium ng isang babae na fetus, okay, take note, fetus pa lang siya. Yung oogonium na yon ay mag-undergo ng mitosis to produce primary oocytes. Next, yung primary oocyte na yon ay isasalang sa meiosis 1, and remember, the products of meiosis 1 are isang secondary oocyte at isang first polar body. However, mayroong plot twist dito, guys. Yung primary oocyte kasi hindi siya magpapatuloy through the meiosis 1. Instead, i-arrest siya sa isang stage ng meiosis 1, and that stage, according to Junquera, is prophase 1. But sir, previously we discussed that prophase 1 consists of 5 stages: leptonema, zygonema, pachinema, diplonema, and diakinesis. So alin sa lima na-arrest talaga yung primary oocyte? Which specific stage of prophase 1? The answer is diplonema or the diplotin stage. But here, since special yung meiosis 1 na pinagdadaanan ng oocyte kasi meron siyang pa-arrest na drama, yung diplonema ay meron siyang special na name dito sa oogenesis, and that is called dictyotin stage. Okay? So the dictyotin stage is equivalent to the diplotin stage. Yung primary oocyte mo na iyon na naka-arrest sa diplotin stage or the dictyotin stage of prophase 1 ay mananatili na naka-arrest sa stage na yon as you grow through your childhood until you reach your puberty. Starting at puberty, at regular intervals, a certain number of primary oocytes will be allowed to finish meiosis 1, producing a secondary oocyte and the first polar body. Paki-disregard lang ng kanang gibutang ni Junquera na meiosis 2 arrested. That should be meiosis 1 arrested. Now, among those several primary oocytes na pinayagang tapusin ang meiosis 1, so mura siya nag-college, no? Isa lang talaga sa kanila ang gagraduate. And that lucky one undergoes a process called ovulation. Ovulation is the process kung saan ina-extrude palabas ng ovary yung maswerteng secondary oocyte na iyon. That secondary oocyte na nag-undergo ng ovulation is the one that will enter the oviduct or the fallopian tube. And it's the one that will fuse with the sperm cell. So, siya yung cell na pinifertilize ng sperm cell. So, remember, that is the secondary oocyte. Ibig sabihin, papasok pa siya, di ba, ng meiosis 2. Yes, papasok siya ng meiosis 2. However, mayroon na namang unique sa kanyang story. Mayroon na namang plot twist. Di ba? Di ba kanina yung primary oocyte, sa anong stage nga siya inarrest? Sa prophase 1. Aling specific stage ng prophase 1? The diplonema or the diplotin stage or the cryotin stage. Itong secondary oocyte na naman na ito, i-arrest na naman siya during metaphase 2. Not anymore in the prophase, but in the metaphase. Metaphase 2. So what stimulates the secondary oocyte to continue through meiosis 2? What will cause the secondary oocyte to continue or to finish meiosis 2? The stimulus is fertilization. Diba? So the fusion of the sperm cell. Ibig sabihin, as long as hindi pa siya na-fertilize ng sperm cell, mananatili siyang secondary oocyte. Now, what if mayroong sperm cell na nag-combine sa kanya or nag-fuse sa kanya? Then that's the time na magde-divide na siya. That's the time na tatapusin niya na yung meiosis 2. So, papasok na siya sa anaphase 2, then telophase 2, then cytokinesis to produce the ovum and another polar body, the second polar body. The next thing that will happen is mayroong mapo-form na zygote which is already diploid kasi yung sperm cell haploid, yung ovum haploid. So, the fusion of the two cells restores the diploid number which is 46. The zygote is diploid, then the zygote will undergo mitosis to produce the embryo and then later fetus. But before we continue through those developmental stages, let us first discuss the hormones that regulate gametogenesis. I got this illustration from Tortora. Let's begin with the hypothalamus. The hypothalamus produces GnRH or gonadotropin releasing hormone. That hormone from the hypothalamus will reach the anterior pituitary, and if you remember your lessons from histology, the specific cells in the anterior pituitary that are stimulated by GnRH are the gonadotrophs. The gonadotrophs will produce the gonadotropins, which are of two types, the FSH or follicle stimulating hormone and LH or luteinizing hormone. Yung gonadotropin releasing hormone produced by the hypothalamus is tinawag, kaya nga siya tinawag na gonadotropin releasing hormone kasi naman yung action niya is sinestimulate niya yung pag-release ng gonadotropin, which is rinirelease naman ng anterior pituitary. Which specific cells of the anterior pituitary? Sagot, gonadotrophs. Now, yung FSH and LH na iyon na pinuduce ng anterior pituitary ay mag-circulate sa blood. Eventually, yung luteinizing hormone molecules ay darating sa Leydig cells. What's the other name for Leydig cells? Interstitial cells. The interstitial cells possess the receptor for the luteinizing hormone. Once the luteinizing hormone binds to its receptor in the plasma membrane of the interstitial cell, the interstitial cells will be stimulated to produce testosterone. Now, the actions of testosterone are the following. Pakibasa na lang yung nasa violet na box. But yung relevant talaga sa discussion natin is testosterone promotes spermatogenesis. Now, another gonadotropin na produce ng anterior pituitary is the FSH. The FSH together with the testosterone promotes spermatogenesis. And the environment that is suitable for spermatogenesis is provided by the sustentacular cells or the Sertoli cells. Sertoli cells will produce inhibin. Inhibin as well as testosterone have a negative regulatory effect on the production of GnRH and the gonadotropins. Now, let's talk about the regulation of oogenesis in females. Just like in males, the process starts with the hypothalamus which produces GnRH. GnRH reaches the gonadotrophs in the anterior pituitary, and then these gonadotrophs in turn will release FSH and LH into the blood. Just as the name suggests, FSH, follicle stimulating hormone. So it promotes the maturation of follicles which contain your oocytes. As the follicles mature, kasi nga gina-stimulate sila ng follicle stimulating hormone, as they mature, they produce moderate levels of inhibin and estrogen. Inhibin and estrogen both have a negative regulatory effect on the production of GnRH and FSH and LH. However, since palaki yung mga follicles sa ovary, darating yung point na napakaraming estrogen na yung pinuproduce nila. Napakataas na ng level ng estrogen. Yes, moderate levels of estrogen have a negative regulatory effect on the production of GnRH and the gonadotropins, but paradoxically, high levels, not moderate but high levels of estrogen, again, what causes the production of high levels of estrogen? Pag nag-mature na masyado yung follicle. Kasi pag nag-mature na kasi yung follicle, marami ng cells ang nagpo-produce ng estrogen, okay? The specific cells kasi ng follicles na nagpo-produce ng estrogens are the granulosa cells, which I believe familiar kayo sa granulosa cells kasi na-discuss to in your histology lab. So yung granulosa cells ay magpo-produce ng napakaraming estrogen. Tumataas na ngayon yung level ng estrogen, and then that high level of estrogen causes a positive feedback loop. Ibig sabihin, mas lalo niyang pinapataas ngayon yung production ng GnRH. The high production of GnRH by the hypothalamus stimulates the production of large amounts of LH by the anterior pituitary. And that phenomenon, we call that the LH surge. Now, the LH surge is the one that causes ovulation. Again, ovulation is the process whereby the oocyte is extruded from the ovary into the fallopian tube. Another job of the luteinizing hormone, aside from causing ovulation, is to stimulate the production of corpus luteum. Kaya nga siya tinawag na luteinizing hormone, luteinizing, because it causes the formation of corpus luteum. Luteinizing, luteum. Hi, finally we're now done with the hormones. But before we proceed to our next topic, let us have a review just to make sure na hindi pa nakalimutan yung napag-usapan na so far. Again, in the seminiferous tubules of the testes, you have three main important cells: the Leydig cells or interstitial cells which produce testosterone, the Sertoli cells or sustentacular cells which produce the process of spermatogenesis, and then you have the developing sperm cells or the sperm cell lineage. In the sperm cell lineage, we begin with the spermatogonium. We have two types, type A and type B. Yung mga type A na stem cells, they replenish themselves, and they also produce the type A na progenitor cells, which will give rise to the type B progenitor cells. The type B spermatogonia will undergo mitosis to produce the primary spermatocytes, which are still diploid, just like the spermatogonia. Then the primary spermatocytes will enter meiosis 1, producing secondary spermatocytes that are of equal size. Okay? I would like to emphasize, equal yung size. Kung ikukumpara mo naman ito sa oogenesis, di ba, iba-iba yung size ng daughter cells. Meron sila mga polar bodies. And then yung secondary spermatocyte na mga haploid na sila, by the way, will enter meiosis 2 to produce spermatids, which are also haploid. Now, those spermatids are still immature. They still need to undergo another round of maturation, and we call that process the spermiogenesis, ending up with those mature sperm cells. Ayan, another illustration from Tortora. Kamo na lang bahala ang mag-go through, kay mas kaya ako nabaog na po ko paulit-ulit. Napulan na po ko, guys. Okay, so babae na punta. In this illustration, we begin with the primary oocyte, which is again arrested at prophase 1. At the dictyotin stage or diplotin stage of prophase 1, at regular intervals starting at puberty, a select number of primary oocytes are allowed to complete meiosis 1 to produce a secondary oocyte and a polar body, which is not included in the figure here. Now, the secondary oocyte will be arrested at metaphase 2. Then it will be it will undergo ovulation kung siya ang mapili na swerte na secondary oocyte. It will undergo ovulation, it will enter the fallopian tube or the uterine tube or the oviduct, and then it is here in the oviduct kung saan siya ma-fertilize. The fertilization by a sperm cell is the stimulus that will allow the secondary oocyte to continue through meiosis 2, producing the ovum and another polar body, the second polar body. Normally, the sperm cell fuses with the secondary oocyte. But what happens if the sperm cell happens to fertilize the polar body? The result is a blight ovum. Bale, lalaki siya, but hindi siya magde-develop into an embryo or fetus. Eventually, this blight ovum will just be eliminated from the uterus. No, i-discharge lang siya. Langman, our reference material for embryology in medicine, enumerated the 3 phases of fertilization. Sir, nga na mag-apil-apil mo kag Langman nga pang-medicine man na. Sa ito dyan, ramata diri sa medtech. Actually, your book, which is your Lewis, also enumerated the 3 layers that your sperm cell has to penetrate to fertilize the ovum. It's just that Langman enumerated these 3 layers concretely, kaya mayroong phases 1 to 3. But definitely, you still have to memorize the 3 layers. Ganun pa rin naman, which are still mentioned in Lewis. Anyway, in phase 1, the sperm cell penetrates the corona radiata, which is the layer of cells surrounding the zona pellucida. In phase 2, yung zona pellucida na naman yung ipepenetrate niya. Remember, kanina we discussed the parts of the sperm cell. Mayroon siyang special na organelle that we call the acrosome. The acrosome contains enzymes that will digest the zona pellucida so that it is able to reach the ovum. And then finally, in phase 3, yung plasma membrane ng ovum at yung plasma membrane ng sperm cell ay mag-fuse. Usually, yung tail ng sperm cell will be left behind. What enters the ovum is only the head of the sperm cell, so that the genetic material contained in the nucleus, which is present in the head of the sperm cell, is able to enter the ovum. And then later, ang next na mangyayari is magko-combine na ngayon yung genetic material ng ovum at saka yung genetic material ng sperm cell. So that nare-restore na ngayon yung diploid number, which is 46. The resulting cell is called a zygote, which is diploid. The zygote will undergo a series of mitotic divisions that we call cleavage. Remember sa previous lesson, ano nga yung dalawang main purpose ng cell division? Reproduction and multicellularity. So doon tayo sa pangalawang purpose, multicellularity. So the zygote is able to give rise to our bodies, which is composed of trillions of cells because of mitotic division. So yun, multicellularity. The daughter cells produced from the zygote are called blastomeres. The blastomeres will form a solid ball of cells which we call the morula. Morula is derived from I'm not sure if Latin or Greek word. Morula etymologically means mulberry kasi the solid ball of cells looks like mulberry naman talaga. Okay, so yung morula, di ba, solid ball siya. When you say solid ball, walang hollow inside. But later on, a space will develop inside. The space inside is called the blastocele, and the stage of development is called blastocyst. Okay, ulit, review. Again, the zygote will undergo cleavage to produce blastomeres. Take note that at this stage, nasa loob pa sila ng zona pellucida. Now, those blastomeres will form a solid ball of cells that we call the morula, still inside the zona pellucida. Later on, mayroong space na magde-develop sa loob ng morula, and that space will be called the blastocele, and the entire stage is called the blastocyst. Which is now outside or nakalaya na siya from the zona pellucida. In the blastocyst stage, mayroong dalawang cell populations. The cells that form the periphery of the blastocyst constitute the trophoblast. The yung cells naman that aggregate inside the blastocyst, we call that the embryoblast, aka inner cell mass. The embryoblast or inner cell mass is the one that will give rise to the embryo. It is the blastocyst that implants into the wall of the uterus. The process in which the blastocyst attaches to the wall of the uterus is called implantation, also known as nidation, Nidation, implantation or nidation. Again, the embryoblast or the inner cell mass is the component of the blastocyst that will give rise to the embryo. So, when can we call the developing human an embryo, and when can we call it a fetus? The boundary is 8 weeks or 2 months. So 8 weeks and below, we still call that the embryo. Then 9 weeks until birth, or until before birth, right before birth, that's when you can call the developing human as a fetus. So yung 8 weeks yung boundary nila. Oh, you don't have to memorize this brochure, ha. Nagbutang-butang lang gid dira para na ko mabutang sa PowerPoint para ano, gi-chargeran ko ra na para lang gid na ma-paste. Anyway, that's all for our discussion. Thank you for your patience. Thank you for listening. Study well for our quiz next Saturday. So these are my references. Medyo daghan kong references. Usog yun sa cytogenetics class kay mag-cross-reference ang teacher. No, we do not settle with just one reference. Unlike during histology na ano lang, hongkera lang sapat na. But ayun, there is a cytogenetics kay sige gid ta og cross-reference because a single book cannot provide all the information that we need to know. So, ayan, happy learning!